Hello and welcome to the pharmaphorum podcast. I'm your host, editor-in-chief, Jonah Comstock. Today, we're going to be talking about cancer vaccine technology. Specifically, we're going to be talking with the leading team from Transgene. I have with me Transgene CEO, Hedi Brahim, and Chief Scientific Officer, Quéméneur. It's so great to have you both on the show.
Hello, Jonah, and thanks a lot for having us today.
Hi to everyone.
So, Hedi, why don't you tell me a little bit about Transgene and how you guys fit into the pharma space and this cancer vaccine space, and a little bit of your history there.
Sure. So we are a biotech company focusing on oncology, and our mission is really to bring new therapies to cancer patients. And our technology is unique because we are using viruses to attack cancer. So we have two platforms, that cancer vaccine and oncolytic viruses, and I'm sure we'll go into more detail about the programs. So we have a deep understanding of the science to optimize viruses to benefit to cancer patients, and we develop them, and then we manage clinical trials to prove the benefit it brings to patients. And we have 4 products that are in clinic right now.
And Eric, do you want to talk a little more about the kind of scientific underpinnings?
Oh, yes. So the idea of using viruses, of course, is to exploit their biological properties. There are many properties that we can use to make drugs out of viral vectors. The first one is, of course, to rely on their immune properties, the fact that they could mobilize at large the innate immunity, boost the innate and adaptive responses, and we have a good knowledge and we can manipulate those responses by engineering the viruses. The second point is that we can use those viruses to deliver to the lymph node or to the tumor very large payloads like large antigens, cytokines, or whatever boosters might be helpful for the anti-tumor response and drive the immune system against the tumor. And I would say second is that we have a very good understanding on our viral vector in order to develop safe but efficient technologies.
So let's lay a little bit of groundwork about this field, and then we can talk about some of the new stuff you guys are working on, including some of your data and your presentations out of ASCO last month. But first, when you talk about a cancer vaccine, how is that different than other cancer therapies? And I know it's a fairly relative term. It's something that sort of it's new in that it is only since sort of the 90s. So how did we kind of discover this space, and where did it start?
Yeah, no, you're right that viral vector-based approaches are considered today as immunotherapy, part of this vast world of different classes of molecules and biological drugs, also part of a very diverse mode of actions. Basically, we could differentiate large molecules that would act on by boosting or repressing some pathways. That is the case, for example, for cytokines or immune checkpoint inhibitors, those antibodies that have been largely promoted in the last decade. There are also many compounds with diverse and very large mode of actions, like cell therapies or viral vectors that would act in a broader manner in the immune system. At the same time, boosting, repressing, and mobilizing many pathways and components. So I can say that there are antibodies, there are cytokines and immune boosters, and there are larger active principles like cells. I think most of your audience would know the CAR-Ts and their progress, but viral vectors have been in the place from the very beginning.
And Hedi, maybe you can talk to me a little bit about from how long have you been with the company, by the way?
I joined the company 18 months ago.
Okay. So you're familiar with the history before then, but not firsthand.
Definitely, yes, exactly.
So when we talk about a vaccine in the normal world of drugs, the difference between a vaccine and a therapy is that you give a vaccine to healthy patients and it keeps them from developing it. Is that how we should think about cancer vaccines? I know like with the HPV vaccine, that's how that sometimes works. Or is it more of a vaccine as therapy kind of situation?
Yeah, excellent question. So it's a vaccine, and because it's going to train the immune system to attack cancer. And you're right that the most known usage of vaccine is to prevent and to train the body before the disease arrives. For example, we train the immune system before we are confronted to COVID, and when we have the virus in front of us, then we are much stronger and we get rid of it easily, and we don't suffer and go to the hospital. Here, our drug is a vaccine because we train the immune system, but it's a therapeutic vaccine because we come after the cancer is here, and we want to help and to restore the capabilities of the immune system to attack the cancer and to get rid of it. And it's interesting that you mentioned HPV vaccine, because indeed they are what we call prophylactic vaccine. And there are large promotion campaign to get young girls and young boys vaccinated. Unfortunately, a lot of people don't get vaccinated, and people can get cancer induced by the papillomavirus. We know a lot about cervix, but there are also other ones. And we have a product that is specific and that comes at the therapeutic vaccine when unfortunately the cancer is there, and we help the immune system of the patient to fight the cancer.
So tell me a little bit about what you guys are working on right now. I know there's a novel personalized cancer vaccine, TG4050, that you guys was your main development you were talking about at ASCO. Talk to me, and I'll let either of you take this, or maybe you can trade it off. But talk to me a little bit about what that is and why it's why it's novel, I guess, and different than what you guys have had before and what's been in the space before.
Sure. So I've just mentioned our product against the papillomavirus. That's TG4001, sorry for the code name. And the newest generation indeed is TG4050, about which we had a poster at ASCO. It shares the same philosophy. It's a therapeutic vaccine. So we come when the disease has already occurred to try to get rid of it or to prevent it to come back, in fact, here. What is very unique about TG4050 is that the product that will be unique for each patient because it's specific to the mutation of the cancer of the patient, and we will train the immune system of the patient against the specific characteristics of its own cancer.
Wow. So how is that accomplished?
Yes, a lot of science, a lot of work behind to make it smooth for the clinicians and the patients. And the process is that there is a tumor sample that is taken in the hospital, a biopsy, so during surgery. Then we look at the genome of the tumor, and you know that tumor cells used to be healthy cells that have accumulated mutations in their DNA that made them abnormal and create this cancer. So we identify all the mutations, and there can be thousands of them for a given cancer. But only less than 1% are immunogenic, meaning that they can be recognized by the immune system. So we look for that needle in the haystack. And for this, we use artificial intelligence because it's impossible to do that by hand, especially if you want to do it for a large number of patients. So we have an artificial intelligence that is designed, is managed by NEC, our Japanese partner, that will give us the most relevant mutations. Then we will manufacture a virus that is coding for this mutation, unique for each patient, and then it's sent to the hospital for treatment.
What's the clinical workflow if you guys are doing this on a patient by patient basis, and how do you make it something that's scalable enough to be, I guess, profitable or sustainable?
Yes, great question. Today we are in clinical trials. We are still in phase 1. We are really delivering a plan in terms of lead time and so on. We are already working on the plan to escalate it to phase 2, then phase 3, and even on commercial. So we know that there are already some individualized therapies. Eric mentioned CAR T cells. Ours is much easier than CAR T cells, for example, to manufacture for different reasons. What is very important also is that it needs to bring a strong benefit. Since it's personalized, since it will be still more complex than something off the shelf, it needs to bring a big difference. And we are very happy of the first science activities we've seen, especially the one we've communicated at ASCO a month ago.
So, Eric, I'm going to toss one to you about immunotherapy and cancer vaccines more broadly. Cancer attacks the body in a specific way that makes it hard for the immune system to respond, the way that it responds to things like the common cold or flu or something, right? It has people talk about stealth cancer and ways that it sort of evades and hides from the immune system. Can you tell me a little bit about that and how you guys go about overcoming that and developing something like this?
Oh, you're right that in our generation, we all learned that cancer should be invisible to the immune system. Now the molecular reasons are well known, and everybody has in mind the Nobel Prize from James Allison and others who have discovered the clues for this invisibility, and we know how to revert that. Reversion can come, and that's the mode of actions of ICIs, those inhibitors of checkpoints that you can just block with anti-PD-1 or anti-CTLA-4. The other way to have the cancer detected by the immune system is to come with a broader and more efficient T-cell repertoire. And that's really what the vaccine is aiming at doing, to boost the diversity and the number of relevant T cells. And so when we design the TG4050, we are particularly cautious in having a large diversity of mutations that we know are tolerated or edited by the immune system. And so we really want to avoid those resistant mechanisms. The idea of new vaccines is really to break tolerance and have the cancer visible again by the immune system. And regarding the ability to have a large immune engagement, that's the reason also why we think that at some time point we might have to combine with immune checkpoint blockers to further boost de novo generated T cells.
So, how does Transgene kind of stand out among other cancer vaccines that are in the market and development, things like BioNTech?
Yeah, okay, you're right. These are the names of the competitors, and basically there are two technologies today. In fact, there are three technologies in competition: mRNAs, viral vectors, and long peptides. BioNTech would be in the field of mRNA. The technology has been largely boosted by the COVID period, and for sure, in terms of manufacturing knowledge on the immune mode of action of their technologies, they have progressed a lot, and they are really key competitors to us. I think that the viral vectors still have a room, in particular for their ability to carry large antigen payloads. Our product is based on 30 different neoantigens. So we cover a large clonal diversity for the tumor mutational burden. And I think that, and we have still room left for more if needed. So in terms of diversity of the presented antigen repertoire, viruses might be better than mRNA. In terms of ability to induce different cell responses, from innate immunity to adaptive immunities, and among them CD8, CD4, and the balance between those two types of cells, I think viral vector have specific properties that differ from mRNA. Too early to say who will be the big winner, but I think there is room for both technologies. We are in fact discussing quite often in conferences with BioNTech colleagues. And as you might have seen, we are targeting sometimes different indications, different stages of the disease, where one or the other technologies might prove some benefits. But I don't think that there will be a one-take-all winner. I think there's room, and there are so many specificities and features for the cancer. And for sure, as we're doing with minimal residual diseases in our preventive vaccines for relapse, I think we are well suited with viruses to do that.
Thanks, Eric. Hedi, what is the scope of this, I guess, this particular 4050, but also your portfolio more broadly? Like, are we talking about one cancer type? Are we talking about multiple cancer types? What's the impact of this?
You know that when you develop the product, you really have to find a particular setting to prove the added value of the product. And then when you've demonstrated it, then you would go and expand in other indications. So we are focusing today for TG4050 in ovarian cancer and head and neck. We are also in gastrointestinal, in melanoma, to name just a few indications. But our technology is not specific to a specific cancer mechanism. We don't target like a specific marker on the cancer. It's much more broad. So we hope we can really prove it brings a different to patient indication that I've named, but typically TG4050 because it's unique for each patient. In theory, it could address most of the cancer type.
And what about stage? Is this something that's going to be more effective at the early stage? Would you combine it with other therapies in a later stage, or how does that work?
Yeah, we've addressed quite a specific need. In ovarian cancer, for example, after first line, most of the patients would have a complete response, but after 1 year, half of them would have a new relapse, which is really dramatic. This very high risk of relapse, and there is no treatment today. So we come after the first line to prevent this relapse. This one women out of 2 to have this relapse after 1 year. So there is a strong medical need, and we think this setting is ideal for a vaccine to train the immune system to get rid of or to prevent the cancer to come back and to get rid of the last traces of cancer that could still be there.
That's really interesting. And I remember there were some other papers at ASCO along the same lines. I mean, this technology really feels like it has the potential to change cancer for something that's chronic and never really gone into something you could really potentially be. Do we use the C word?
Yes, definitely, and really we are addressing like new stages of cancer. Today there is an emerging technology that is ctDNA, circulating tumor DNA, that you can have a blood sample and you quantify the number of DNA from the tumor that is in the blood. And if there is a lot, it means the cancer is massive and coming back and aggressive. And if not, it's reducing and maybe disappearing. So that is what we are tracking, and we can attack cancers at very early stage and even prove that we have an impact, even it's sometimes not even detectable by any imagery system.
Wow. We're coming towards the end of our time. Is there anything either of you want to address that we haven't talked about yet? Do you think is important or is interesting?
Just one word, we really focused on therapeutic vaccine. We're very proud of the 2 products. But we also have another platform that is oncolytic viruses. It's another kind of viruses that will directly attack the cancer cell. But I think we need another discussion, 20-minute discussion to go through that for us another day.
Interesting. And so is that those therapies kind of complementary to each other?
Yes, they attack different needs, different stages of cancer, and a really different mechanism, even if it's still viruses.
Eric, any other thoughts from you?
No, I think maybe in a nutshell, I think that we are happy with those programs that we have described to have viral vectors entering in the field of precision oncologies, combined with AI, tumor genomics, ctDNA, liquid biopsies. We are surrounded by the arrival of the new technologies that bring a lot in terms of deep knowledge on specific patient characteristics, and immunotherapy and viral vector-based immunotherapies would be ready for that.
Great. Well, thank you both so much for joining me. Really, really fascinating stuff and very exciting. What are, just to finish off, what are you guys excited about for the future, looking ahead in this technology?
Thanks, Jonah, for your time. But a lot of milestones, of course, short term for the programs with readouts and so on. Really excited. For example, we just discussed about TG4050. We'll have a new poster at a new major congress before the end of the year. So really, today we know we are still in phase 1 or phase 2, so it's a limited number of patients. But if we can make a difference for these patients, that would be a really great for them and for us, and very promising for the future.
Awesome. Thank you all so much. I think this has been a great interview, and look forward to hearing more.
Thanks, Jonah. Bye.
Thanks, Jonah.
That concludes this episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about other installments in the series, at pharmaphorum.com/podcast. The pharmaphorum podcast is also available on iTunes, Spotify, Acast, Stitcher, and Podme, where you can find and subscribe by searching for pharmaphorum. And don't forget to visit our website, where you can sign up for daily news and analysis bulletins, and to follow us on Twitter at pharmaphorum. Thanks for listening.